Welcome. I am Meg Smith, CEO of Cure Alzheimer's Fund. Thank you for joining us today to hear about how we are advancing the fight against Alzheimer's disease. Cure Alzheimer's Fund has one mission: to fund research with the highest probability of preventing, slowing, or reversing Alzheimer's disease. Since 2004, we have deployed more than $210 million toward extraordinary research to end the terrible burden of this disease. Active researchers from a wide range of scientific specialties who are at the forefront of discovery guide our funding while also pursuing their own breakthrough projects with our support. At CureAls, we focus only on making bold bets to generate knowledge that will help the entire Alzheimer's community. We do not pursue profits or financial returns. We can provide this support to the most innovative ideas across biomedical science, prioritizing new, neglected, and unfunded questions fundamental to understanding what causes and contributes to Alzheimer's, only because of the generosity of our donors. Our board of directors, trustees, and certain other supporters cover all of our operating costs. This generosity and vision mean that 100% of the gifts from our other wonderful donors go directly to our research program. We are grateful for the community of generous donors who make these scientific advances possible, and I am glad to share some of them with you today. After watching, I hope you feel as I do: incredible resolve to find a cure for everyone touched by this terrible disease.
Thank you so much for giving me the opportunity to speak to you today. Cure Alz is my favorite group of people. Fantastic management, fantastic science, fantastic interactions, really absolutely the best. I couldn't wish to be part of a better group. Anyhow, so having said that, I'm going to give you some background on me and how I came to be in this position to be able to talk to you today about the research we're doing, and framing it with some of the history of the field that I've been involved in since 1992, and sort of finishing up with some of the questions that we want to answer that we feel are outstanding questions in the field. And with the help of Cure Alz, how we're going about answering those questions. So I'm currently in London. I'm the Centre Director of the UCL Dementia Research Institute. And up until 6 years ago, I was at Columbia University, and I was in the States for 27 years prior to that. So I'm fairly well versed in some of the American terms, so hopefully I won't switch between English and American too much. Anyhow, I thought we'd talk about tau. Tau is the field I've been working in for about 30 years. But really the question that we have is now that we have reasonable immunotherapy drugs and a lot more drugs in the pipeline that will target A beta and amyloid, the question is really, is this enough? Has all the work that we've done over the decades leading up to this point, where we have in clinic some pretty reasonable drugs or therapies that will reduce A beta and amyloid, is that going to be enough? Or are you really going to not see much of a shift in the dial of trying to cure or prevent Alzheimer's? So I started, just a little bit of background here. So I did my PhD in 1991, and I worked with the great Sidney Brenner, who won the Nobel Prize for his work on RNA, and I worked at Cambridge. And my thesis actually wasn't on Alzheimer's disease, it was on Down syndrome. And Down syndrome is actually an anomaly caused by triplication, 3 copies of one of the chromosomes, chromosome 21, which is very important in Alzheimer's disease, and I'll show you why later on. But my question was really, how do genetic deficits cause complex diseases like Down syndrome? And latterly, I worked on Alzheimer's disease. So I've always been fascinated by these single mutations in the DNA and how they give rise to a very complex disease later on in life. And this is Sidney Brenner. He was quite the character, and he had many quotes, but I thought this one was quite funny because science depends on new techniques, new discoveries, and new ideas, and probably in that order. And I actually couldn't agree more with him that we always sort of create new ways of looking at things, and then we sort of often retrofit a question to the techniques that we've developed. So after my PhD, I was thinking more about chromosome 21, and I was thinking about Alzheimer's disease. Now, Alzheimer's disease at that time in the early 90s was well defined by pathology. And here you have the classic pathology of Alzheimer's, the amyloid plaque, which is caused by the accumulation of the A beta peptide. And this comes from a gene on chromosome 21, which was my Down's syndrome chromosome. So I was immediately linked with Down syndrome and Alzheimer's disease. And sure enough, a lot of Down syndrome people get Alzheimer's disease. And it's because of this link to the gene that's on chromosome 21 that causes amyloid plaques to form. But in addition to amyloid plaques, you have these neurofibrillary tangles. So Alzheimer's has always been known to be a disease with two pathologies. But the amyloid, the predominance of amyloid, has ruled the field for decades. And I'll talk a little bit about why that is and why that should continue to be the case, or whether we need to really think carefully about what other causes we should be targeting therapeutically. So in addition to those two pathologies, you have a big inflammatory response with astrocytes and microglia cells, and you have the loss of your cell connections, your synapses, and the degeneration of the neurons. And of course, this results in the cognitive impairment, the memory loss, the loss of ability to interpret the world and know who your loved ones are, that we all is so traumatic for those with Alzheimer's and those who care for them. So the predominant hypothesis at the time was this, the amyloid cascade hypothesis. And this was based on this idea that the amyloid plaques were very much the driver of everything that happened in the brain that caused the dementia. And this was thought to go through something related to the generation of the component of the plaque and the tangles forming, and then the cognitive impairment happening after that as a result. Everything upstream being caused by something to do with amyloid and A beta. And a lot of this was proposed in the early 90s by this man. This is John Hardy, now Professor Sir John Hardy. He was knighted. And he really started the ideas about the amyloid cascade hypothesis, working together with Alison Goat, who's now at WashU, who's now at Mount Sinai in New York, having spent several years at WashU. So these two together identified mutations, and here it is, first 8 mutations in the amyloid precursor protein in 1991. This was the actual gel. These are all the little bands showing DNA bases. And when you have a genetic mutation, one of these is wrong. It's not the usual CAGC that you would expect. It's wrong. And this was the actual first glimpse into the genetic cause of Alzheimer's disease in a patient in a family in London in the early 90s. And this led the way for this amyloid cascade hypothesis, which follows this protein called the amyloid precursor protein, APP, through its journey being clipped and cut and coming together, clumping into the brain in the amyloid plaque that we know from the pathology. Now, there are several events, and I won't go into them in too much detail, except they're very, very important. They're very important for targeting with therapies. They're very important for understanding what causes Alzheimer's disease, and it's really fundamental to understand this process. And a lot of work has been done to do just that. The first mutations were here in one end of this amyloid precursor protein, and the second set of mutations were in another end of the amyloid precursor protein. And it clearly showed that cutting out this protein into this little chunk here, which then went off and formed into the amyloid plaques, was causative of Alzheimer's disease in these cases with this familial form of the disease. So I was working with John Hardy, and I think you can see this is a very early picture of myself working with John Hardy. At this time, we moved from London to the University of South Florida. He took up a professorship. I was a postdoc in the lab. This is me in 1992. And in this lab, during this time, we discovered the second set of genes that caused Alzheimer's disease in familial cases. This is the presenilin gene. And I was the first author on the mouse model that we created to look at how these proteins interact and cause a disease. So the fact that you kept finding these mutations in the amyloid precursor protein were really fundamental for how we thought about the disease and how, especially, we thought we were going to cure it. And this idea that everything to do with curing Alzheimer's disease would rely on the cutting up of the amyloid precursor protein into these little bits that went and formed the amyloid plaques. This was thought to be the only way you were going to cure the disease. And it to this day predominates as the mechanism of choice for therapeutic approaches. And the lecanemab that you have in the clinic and the other immunotherapies and the secretase therapies, they're based on this idea that removing parts of amyloid and A beta are key to curing or preventing the disease. So after my stint at the University of South Florida, I moved to the Mayo Clinic. I moved with John Hardy again and some of the other team who I'd come over from London with, and we moved to the Mayo Clinic, where I started to try and generate really good mouse models for the disease, because you need a mouse model to be able to model the disease so that you can test your drugs on it. So my job was really to use genetic engineering to create the best mouse models of the disease that we possibly could. And this resulted in putting together different mutations and making a more powerful mouse. And we were able to create one of the best mouse models for drug studies in the early mid-90s. But I was really curious at this point, because Alzheimer's disease is a disease with two pathologies, as I mentioned, not only the amyloid plaques, but these neurofibrillary tangles. And everybody was sort of ignoring this because they all thought APP and amyloid was the only way to get at the disease. I was like, well, we haven't got a full model of this disease. We don't have the plaques and the tangles. So we wanted to create these mouse models that had the tau pathology so that we could actually put together the amyloid pathology and the tau pathology in a Alzheimer's disease. So this mouse that we made in the early 2000s modeled the tau pathology pretty well, but it wasn't a model of Alzheimer's disease. I dedicated the rest of my early career to looking at everything to do with tau pathology and tau biology. And I moved up to the Nathan Klein Institute, part of NYU, in the late 1990s, where I started to look at different reasons why the tau protein would go astray, right? So I'm looking at what causes the tau pathology to develop. At this point, I had two daughters, which was great in the early 2000s. And I also moved to Columbia University Medical Center in 2006. And here I wanted to start to think about some of the aspects of tau pathology that intrigued people for centuries, in fact. This was one of an early observations about tau pathology, that it started in one area of the brain, but it spread through the brain. Suddenly, later on in the disease, it was seen in different areas of the brain to where it started. And this was a conundrum. How did the tau protein do this? Why did it go from one region to another over time? Was this involved in some mechanism that we could actually prevent? We could target the tau and stop it in its tracks before it could infect the whole brain. So this started the whole idea about tau spread and the spread of tau pathology. And this was sort of one of the things that I became fairly well known for, partly because I made new mouse models where I could look at tau pathology in areas and see with time why it spread and infected new areas of the brain, as was shown in this mouse model. We published multiple papers looking at this and also trying to think of how we could therapeutically catch the tau as it was spreading from one cell to another. And we continue to this day to look at how tau spreads from one cell here, which would be down here, to another cell, say a cell up here. How does it do that? Does it jump? Is it actual tau pieces that float across from one cell to another? Are they within little encapsulated forms? And how can we actually capture that tau and take it out so that it's not going to form a tangle? So we spent a lot of time looking at that. We also were looking at another feature of the disease which intrigues people, and that is, why does a tau tangle only infect some neurons and not neighboring ones? Why is it selective in where it locates and accumulates? Why is it only in this cell and not this cell when they're right next door to each other in the brain? Every cell is not vulnerable to accumulate the tau pathology. And the idea here is, why does that happen? But secondly, how can we boost the resilience of a cell to not be vulnerable to accumulate this tau pathology? How can we actually prevent a cell from developing tau pathology? And this opened up an entire new area of looking at cellular vulnerability and resilience and how we can boost that. So I then moved to London, 2019, and I'm the Centre Director at the DRI, as I said. And my work has continued on many levels. We look at clinical and genetic diversity, different genes, how they cause diseases. We make new mouse models, so we have better mouse models of diseases. We are developing cell models that are better models of the human brain, and using fairly sophisticated new cell models for that. We're using cell models that are derived from human patients so that we can better model the disease, again, in the cell models. We're using computational approaches and also now AI approaches to try and understand the basis of the disease better. And we're developing better models for the cognitive problems, the memory problems that plague Alzheimer's patients. How can we model those better so that we can understand whether our drugs are actually effective at not just stopping these, pathologies, which are important, but, what's really matters is preventing the memory loss. So how can we develop better cognitive tests for that? We do some drug discovery. We're very interested in turning all this knowledge into new drugs. And we also are very interested in early diagnosis of the disease, because the earlier you can diagnose it, the better you're likely going to be able to treat it. And I just wanted to focus just for a minute on this lovely new behavior test we have for mouse models. And there are lots of deficits, problems in trying to understand the memory of a mouse and whether it's the same as the memory of a person. And we really want to be able to have the right model so that we understand the dementia that happens in people. So we've devised this new test. Sorry, can I take that off? And this is a mouse. He's running on a ball, and he's using virtual reality to understand his environment. This is what our mouse who's here is seeing. He's seeing a track, but this is in virtual reality. He's got a headset on, like your kids would use, when they're doing VR. And he needs to know when a reward is coming. He gets a little milk drop, which he loves. And he needs to know how fast he has to go because he's using the cues on the side. And this memory test is a test of something called spatial navigation. And it's a very early problem in Alzheimer's disease. And you might be familiar with people with Alzheimer's disease very early on get lost in their surroundings. They get confused. They don't know where they are. And so this is a very early sign of deficits in brain regions that we think are targeted by tau pathology. And so we're modeling them in mice with these new virtual reality-based methods and matching them with human. And I just wanted to highlight a family that we have been working with to be able to test these new methods of understanding the dementia. And this is a family that's in Colombia, in Medellin. And they have 6,000 people in this family who have one of those mutations, the presenilin mutation that causes very early Alzheimer's disease. They get it in their mid-40s, and it's absolutely tragic. But this allows us this high number of people with the disease where it's really known when they're going to get the disease, very close age, to the parents who had the disease. So we can actually start to do testing of these type of approaches. And this is my colleagues. This is John O'Keefe, who won the Nobel Prize for spatial navigation in rodents. And my colleagues at UCL, this is me wearing this virtual reality headset. And we're in Colombia developing this test so that we can monitor people very early on and their dementia, and hopefully bring that back as a very early diagnostic test for whether people are on their way to getting dementia, Alzheimer's specifically. We went off to see some of these Alzheimer's patients in the villages where they live so that we would understand the landscape they were living in. Went off in a helicopter. And this was some of the family members who were affected with Alzheimer's disease. And actually, this lady here has Alzheimer's. You can see how young she is. So this is the wonderful Colombian family who have dedicated their lives actually to helping scientists understand the basis of Alzheimer's disease. So there's a lot to be done in understanding different populations and being able to understand not only what causes the disease in these patients, but actually some of them don't get the disease even though they should because they're genetically predisposed. And that idea, why they're resilient, is a whole new field. How are they protected against it? What is protecting them? And we're really interested in that idea about resilience, not just what makes them vulnerable. And I just wanted to end with the work that we're doing with the Cure Alz, Foundation. And this is the Tau Consortium that we put together to really start to understand how Tau is impactful in Alzheimer's disease, and especially what is the relationship with amyloid. We do think that amyloid comes first, and a beta deficit problem comes first, and it drives the Tau pathology. But the Tau is a killer. It will kill neurons. And we know that you're going to have to probably stop the Tau pathology, stop the deficits with Tau to really have a big impact in the disease. So what is the relationship between these two known pathologies? What do we understand the biology of their interaction? We have a couple of goals as a consortium. We want to understand what starts this whole process with the Tau pathology. We want to understand, why does it progress? Why does it jump from cell to cell? Spread through the brain, which makes people much worse. It makes their cognition really bad and their dementia declines very rapidly. We want to understand, why does it look the way it looks and whether we can actually stop it. And the consortium is 5 groups of us from mainly across the US, but also myself and my colleague are here in the UK. Henry Zetterberg's group is in Sweden. Bob Vassar and Catherine are at Northwestern. Brad Hyman and Rachel are at Harvard, and Mark Diamond is in Texas. And together we have a coordinated program to really apply all of our knowledge of Tau, basic Tau biology, but also our knowledge of amyloid and APP biology to understand how these two things come together. We're in the 3rd year of the consortium. We had a very successful first 2 years. We've expanded the program, and we would like to expand it some more because we really feel this is where the rubber hits the road, right? I think you really have to understand this. And classically, all of the therapies, as I said, are targeted against APP and amyloid. We don't have much for Tau, and it really is going to be crucial if you want to make a big impact in the disease. So if you're curious about some of my early life, this is a hilarious pair of articles that were published about me. One is in Lancet Neurology and the other is in the British Neuroscience Association. And you're welcome to have a look at where I came from in the field. This is my professional life, not my personal life, although you can see I quite like riding motorcycles, and I had a brief stint as a motorcycle stunt rider. So of course, people pick up on that. But this is really how I came to be interested in Alzheimer's disease and my 30+ year experience of working in the field and dedication to Alzheimer's disease. So very much Cure Alzheimer's for my funding. It's really been instrumental. I can't say enough about how impactful it is to have this flexible, very informed group of, flexible funding with very informed, sort of insightful people understanding what the important questions are in the disease and how we should really be thinking to be very effective going forwards in how we deal with the disease. So thank you to Cure Alzheimer's and thank you to everybody on the call.
I hope you enjoyed this presentation of some of the cutting-edge research that Cure Alzheimer's Fund supports. I invite you to find a wealth of additional information on our website, curealz.org. Thank you for spending this time with us to hear about the science and progress that will bring an end to Alzheimer's. Please be in touch should you have any questions about the science we fund or how you can be part of the solution.